Okay, I think it's 10 o'clock. So, well, welcome everybody. I don't know where, but anyway, because here in our room there is just about 20 people. So, but I hope there are some more somewhere there besides the computers. So, welcome you from Warsaw, from veterinary faculty in Warsaw, in Poland, and we are together with the dean of our faculty. Just to start this our conference, our symposium. Well, the symposium is organized thanks to IDVET company who supported us just with money. The dean supported us with the technical opportunities, with the room, and the transmission is also thanks to faculty and thanks to the university. Also, the Warsaw Chamber of Veterinary Surgeons supported us. Thank you very much, and I hope they are also some veterinarians from Poland just watching us now. Well, I would like also to welcome the colleagues from the small ruminant from the College of Small Ruminant Health Management. Hello, if you are somewhere here. Well, I would like also to welcome the colleagues from International Goat Association. Hello, hello, I hope you join us also. Well, I would like to welcome also the Polish vets who are members of the Polish Society of Veterinary Medicine, because this society is also helping us to organize the whole thing. And also I would like to welcome the colleagues who are from ICRAT project, because openly speaking, this symposium is mainly organized just to show what we do in an ICRAT project, because our colleague Michał Czopowicz, who will be the last one presenting the results, so he leads the project. They are in between this project. They are also colleagues from Switzerland, from Norway, from Lithuania, and also from Hungary. Welcome everybody. We will have also the colleagues from Romania to present the results, also from Germany, also from France. So I welcome everybody, and thank you very much that you came. And I hope you will have you already have had, and maybe you will have also a nice time here on our faculty and in Poland. Okay, so I think now it's the time just to officially open our symposium. So Michał, please, you are the host here.
Thank you,
You are the boss here, so please.
Thank you very much. It's time for more official way. I'm sorry that I'm using additional materials. Students are not allowed, but as a dean, I can use it. Sorry. It is my great pleasure to participate together with my colleague, Professor Jarosław Kaba, in the...
And your former teacher, unfortunately.
Yeah, and my teacher. I had the best teachers ever.
Thank you very much. Thank you very much. And I have the best students, you know, ever also.
In the opening of the Symposium of Caprine Arthritis Encephalitis, which is indeed demanding disease, both for breeders and veterinarians. It is my honor. It is an honor for me to welcome so many distinguished guests from all over the world. I hope so.
Me too.
And the specialists willing to share their insights on this specific thesis. It so happens that Warsaw has, for so many years, thanks to efforts of professor and his research team and his friends from all over the world, that Warsaw became a leading center in the field of small ruminant diseases. So I hope that today's meeting will be an excellent place for sharing ideas, presenting results of scientific research, and to tight social
which is also
relations, social bonds. Since I'm among passionate people, because you're passionate people, I'm sure that the time spent together will be a source of inspiration for further research, I hope, in international teams. So, in addition, I wish you smile and cheerfulness. Sorry, before the symposium began, I had already improved my knowledge on goats, because until recently I did not realize that in English male goat is referred to as Billy. And young individuals of these species are referred as to kids. It was a surprise for me, because I'm not specialist in this field. So therefore, all of us should be nice to goats and show patience, because they are just kids. Yeah? Enjoy the meeting together and have fun.
Yeah, thank you very much. Thanks a lot. Thank you very much. Thanks one more for the faculty and for the university to help us to organize the meeting. Michał, thank you very much. It was really nice that you came and opened our conference. Thanks a lot once more. Thank you. Thank you. Okay, so we are starting our conference, and as you probably know, we have just 10 lectures. Each lecture will be approximately 15 minutes. Zosia, who is just here, will be watching the time. 5 minutes for the questions and answers. So if you have some questions, please write them on the chat. So then if we have a time, we will try to answer the questions. And we will try to follow the time, but you know, sometimes it can be some delays, but let's hope we will be just on time. Okay, so let's start from the first lecture, and well, this is just as you see me. I would like just to tell you some really basic information about the capran arthritis encephalitis, and well, in fact, I would like just to exchange with you some our own information about the capran arthritis, which we are dealing with over 25 years. Some of those informations, they are, well, all of them, almost all of them, they are just from our researches done during these 25 years. Of course, some of the results are, you can discuss with them, but what we can say, those are just the results from our work. So as you know, for sure, the disease is caused by the small ruminant lentivirus, and there are two genotypes. The genotype A, it is this one, which a long time ago, maybe not so long, but anyway, we called it in the past Medivisna virus, and genotype B, it's this one which in the past was responsible for the capran arthritis encephalitis virus. Now we just say that it's just a small ruminant lentivirus, and of course some basic information about the clinical course of the disease. And well, mostly the infection is subclinical, so there is no any clinical symptoms. This is most often what we see. The animals are infected, but we don't see the clinical symptoms. If they are the clinical symptoms, so most often what we see, it's just arthritis. It's a painful arthritis, and of course the goat simply suffers. Well, many times also we see the losing of the condition, and that those signs can be together. I mean, arthritis with the losing of the condition of the animal, and as you can imagine, of course, the animal is really weak. And if we come to the production. So of course, no doubt, such goat will produce less milk. That's without any questions. Well, in many, we don't see any clinical symptoms from the respiratory tract. But if we do the postmortem, so in something like 40% of the cases, we see the lesions in the lung. Well, sometimes we see also the viral mastitis, which is called by the farmers, usually had other. Well, not very often, but we see it, but very, very seldom we see the neurological symptoms. Openly speaking, well, we dealt already 25 years with this disease, and me, myself, I have seen only one case like that, really. So I really believe that it was capsular arthritis encephalitis. So it's really, really very, very, very seldom. Okay, so I said that mostly we have the subclinical course of the disease, sometimes a clinical, and now it's the question, well, why it's like that? Because, well, we have some goat herds in Poland. We know that they are infected, and we can see almost not any symptoms. Yesterday we have been visiting one farm. They were it was something like 100 animals, and maybe 2 had some lesions, yeah. If we go to another farm somewhere in Poland, so we have the farm with something 100 animals, and let's say 70% of them, they have the clinical signs. So how it is like that? Well, there are some publications that this is just because the genotype A, they are the publications that this genotype A, so this Medivisna virus, is less pathogenic for goats. But, well, from our observation, it's not like that, sorry. This is the herd I just told you about. If you look on those, on the knees, terrible lesions, and also genotype A. So I think the question, the answer to this question is not so easy. We can't say that only the genotype A or B is responsible for difference in the course of the disease. Well, maybe the strain, not only genotype, but maybe the strain of the virus, maybe the genetics of the goats, maybe the environmental conditions in which the goats are kept, maybe all those factors together, maybe something else. Well, who knows? But from my observation, this answer, I mean, genotype only, does not fit to the reality. So I don't think so that this is only just because of the genotype. Okay, so the disease, of course, important. Always we say all the diseases are important, especially those in which we are good. So those are for sure important. But CAE, it is important. And of course, there is not any doubt that, as I already mentioned and show you, those goats really suffer. So no doubt that it has a huge influence on the welfare. Of course, when we have the clinical symptoms, clinical signs, then it's for sure very important. However, we have done such a study in a goat herd were, of course, infected. Not one, in the many goats in Poland. That was epidemiological studies. We just asked the farmers whether they see the problem, I mean, arthritis in their herd or not. We have collected a lot of such data. And, well, what are the conclusions? And then, of course, we did the serology in all those herds. And what are the outcomes? So the owners, we are talking only about the infected herds. So the owners see some problems, arthritis mostly, only when the seroprevalence between the herd is over 40, over 70%. So still, if the prevalence between the herd is lower, so even the owners don't see any problem. Well, the other thing which should be obvious, if it's the culling rate, well, this is also done on the huge population of Polish goats. only those herds which are infected. So you see, this is the percentage, the prevalence of infected animals according to their age. So the highest percentage it was in the goats which were 4 years old. Then it dropped. Why? It is just the time when the symptoms appear, when the signs appear, and of course those goats, they had to be the owners had to get rid of them. So you see, of course, the impact on the lifespan, it's obvious, no discussion. If it comes to the milk production, then again we can discuss it. Well, we did study on the influence of the infection on the milk yield, and we have to say that we couldn't prove it. Sorry, you know, there are some papers, they say it is even big. There are some papers saying that not. Well, we could not prove. Again, why? Maybe again the genotype of the goats, maybe the strain of the virus, maybe the environmental condition, the production system, maybe in our case it was semi-intensive. Well, if we discuss the somatic cell counts, so similar situation, we were not able to prove the statistical influence on the somatic cell count. There are some papers, they say yes, it has. So again, this is something which needs a discussion. We also check how the infection influence the milk composition and proteins, fat, lactose. You see, there are differences. So we could say yes, it is a negative effect of the infection. But if you look on the differences, 0.05%, 0.15%, 0 point. So very low. Okay, it's statistically significant, but whether it's significant from the economical point of view, I don't know. Again, of course it is, but whether important, I don't know. Because well, in Poland we keep the goats mostly for cheese production, so we checked also what's the influence of the infection on the cheese yield, and you see it is okay. But again, if we come to the real numbers, this is 4.6 grams of cheese per 1 kilogram of milk. Again, statistically significant, but from the practical point of view, well, this is the question. Okay, so it is, but, well, it is like it is. Let's go to the reproduction. We did also such study. So the influence on the litter size, no influence. On the weight of kids just after birth, no influence, and also on the weight of the young kids until 3 months of age, no influence. So just summarizing all those things, for sure there is a big, really, without doubt, influence of the infection on the welfare of animals. That's for sure. If we come to the milk production, well, we can discuss it. If we come to the reproduction, rather not. So someone could say, okay, so this is not an important disease, but, well, this is my goat. Well, it was my goat. It passed away already because of the capron arthritis and encephalitis. Look how poor this goat is. So this is how the disease ends. So we can't say that it's not important disease, for sure not. Well, so we should do something, we should act, and well, everybody knows that the treatment is not possible, not any treatment possible, no vaccination possible. So the only thing what we can do, we can just test the animals and just get rid of those which are infected. That's okay. We can try to rear the kids without the colostrum of the mothers and so on. But anyway, everything is based on the diagnostic method. So we really need good, highly accurate, cheap also, because we are talking about the big population of animals, and easy to use diagnostic test. So Well, in my opinion, that's something most important in case of Capran arthritis encephalitis. And well, openly speaking, this conference is mostly just because of this. Because, well, our idea was, and Michał will tell it on the end, our idea was just to try to establish such a test, which will be cheap, which will be easy to use. So someone could just check the animal in the stable, and which will be really accurate. Because in our opinion, that's the most important thing in case of Capran arthritis encephalitis. And this is, you remember this poor goat, the picture of this poor goat. So it's the same goat, just half a year before. It was wonderful goat. It was my goat, my lovely goat. And it took only 3 months, and we had to just put it asleep because it was just suffering. So from one side, I try to show you that this is not very important disease from the maybe economical point of view, but from other side, you know, it's really important, and it's really well, devastating disease. Okay, thank you very much. That's all what I would like to say. Do we have any questions? Okay, so we don't have any questions from the audience, maybe. Not any questions from the audience. Okay, so I would like to. Okay, thank you very much. So Adrian, could you come? So the next lecture will give us Adrien Limousin from IDVET from France. So of course it will be about diagnostic methods.
Of course, yes.
ELISAs. The floor is yours.
Thank you, Professor Kabas, for your warm welcome here in Poland, and thank you for the organization to you and all your team. So yeah, as you mentioned, I'm going to talk about diagnostic and more especially about ELISA. So the title of this presentation is the diagnostic performances of our test, the ID screen, MVV and Capran arthritis and encephalitis indirect screening test. So before I start, I will give you some introduction about the virus, but I'm sure you're all expert on this subject. Then I will focus more on the test and on the test performances, and in the end I will do a little review of what is being said in the literature. So to introduce these two viruses, so here you have a picture, a scheme of the virus with all the proteins that are contained in the virus. These two viruses are very genetically closed, and they are also known as small related lentiviruses, as Professor Kaba introduced you to these virus. Medivisna virus is also known as ovine progressive pneumonia, OPP, and they are both characterized by very long life persistence in host monocytes and macrophage. The infected animals are mainly subclinicals, so there is no evident clinical sign, but these animals are still infected and are able to transmit the virus, so we need clear diagnosis to spot these infected animals. This is a quote from the OIE chapter manuals about this disease, and I found it very relevant. And it said that the most practical and reliable approach to confirm a diagnostic of infection of these two viruses is a combination of serology and a clinical evaluation. So this is also extracted from the OIE, and this is all the diagnostic method that there is available for the diagnostic of these two viruses. And we can see that here there is the AGID, which was historically used to diagnose this disease for all these purposes, for example, to contribute to eradication policies or to confirm clinical cases. But now we can see that ELISA is very popular and can be used for all the listed purposes. So what makes a good ELISA? So the reliability of the ELISA depends on 3 factors. The first one is the assay format. The second one is the homology between the strains that is on the field and that is used in the test. And the third one is the viral antigen that is used in the assay, and I'm going to linger a little bit more on that topic. So here are piece of general information about the MVV and CAE genoma. So there is three kind of component. The first one is three structural gene, the GAG, POOL, and ENV genes. The second one is three accessory genes, VIF, TAT, and REV. And there is also long terminal repetition. And we are going to focus on this first one, the three structural genes. So among these structural genes, there is the GAG, the group-specific antigen, which are very highly conservated in ovine and caprine lentiviruses, and it codes for three proteins. The two first ones are the P14, the nucleocapsid proteins, and the P17. And the last one, the third one, is the P25, which is of very high interest for our test, because this P25 induce a very strong immune response in infected animal and has therefore been used as a basis for the development of diagnostic tests. There is then the second category, the ENV genes from envelope, ENV for envelope. It codes for the glycoprotein GP160, and within the host cell, this glycoprotein is divided into a GP135, the SU protein, the surface glycoprotein, and this is responsible for the interaction of the virus with the host cell receptor, and the GP44, also known as the transmembrane glycoprotein, which is anchoring the first one to the envelope. So these three peptides are of interest because highly conserved and immunogenic in the animals. And this is why we used a cocktail and a mix of these three proteins, so the transmembrane proteins, the GP135 and the P25 proteins, to coat the plates that are the basis of the ID screen ELISA. So let's focus a little bit more on the test and its main characteristics. Let's start by reminding you how it's working, because this test is an indirect one. So on the right side, you have what happened when it's used on a negative sample, and here on a positive sample. So as I mentioned, the three proteins are coated on the plate. Then the antibody, well, the serum is added, and if the antibody is present, it will bind to the antigen coated on the plate. Then you add a conjugate antibody, which will recognize the antibody which is or not contained in a serum, which is binded to the enzyme HRP. And then you add the substrate solution, which will get a yellow color if the sample is positive. And then you will have results with an optical density value. So here is the kit. So it has been validated on goat and sheep. It is used to detect antibody against both viruses, and it can be used on serum, plasma, and milk. Here are the main characteristics. So as I mentioned, this is an indirect ELISA, which can be used on three matrices. The specimen volume needed to perform the test is 10 microliters for serum and 50 microliters for milk. And the test runtime is only 1 hour and a half. So this is a very convenient, rapid, and easy to use test. There is also an associated product, the MRI, which stands for Material Reference Internal, which can be used to validate and to see if the kit is correctly working. I was talking about the result interpretation. You will obtain an optical density, which will be converted thanks to a calculation to an SNP value, which will be negative if this value is below 15%, doubtful if this is between 50 and 60%, and positive above 60%. So what about the test performances? So first, I'd like to talk about the sensitivity on serum. So here you have the results we obtained on 83 EVE milk sample that were naturally infected with genotype A virus. And you can see here in the positive zone that there is no samples that have been detected as negative. And we have a very acceptable margin between the last positive samples and the cutoff of the test. So we obtain a measured sensitivity of 100%, which is very excellent. And this time on milk, so this was on serum, and we also performed some analysis on milk. Here, this is the same logic. We tested on 173 samples from CAE infected herd in France. And one more time, we found every samples positive. So all animal status were obviously confirmed with PCR to be sure that they were real positive and not false positive. So one more time, we obtained 100% of sensitivity on milk this time with the ID screen kit. So what about specificity? So we also tested on serum and milk. So on the right and left side, sorry, this is the test on for specificity on serum on almost 600 sheep and approximately 300 goats from disease-free herds. And here you have the SNP value we obtained horizontally and the percentage of tested sera. And we can see that either for sheep or goats, every negative sample were found really negative with this test. Same logic with milk. We tested this ID screen kit with 132 EVE milk from disease-free herd, and one more time, every samples were in the negative zone. So we obtain, this is with cut, but we obtain 100% of sensitivity and of specificity, sorry, in both matrices. This has been also described in the literature. There is an effect of the BTV8 vaccination on the diagnostic of small ruminant lentiviruses. So we performed a study on 150 serum from animals that were vaccinated with BTV8, and we obtained no positive results due to this vaccination. So this result has been published in a poster that you can see here on the right corner of the slide. But the thing is that there has not been clear evidence of an interaction of this vaccination against BTV when we are using this diagnostic kit to screen the population for this virus. So these data were the data that we obtained internally at IDVET. What about the external references? Because there is also many studies that are using this kit. First, I'd like to start with the performance evaluation on milk. And the first study compared our test with the test from IFEN, the ELI test ELISA. And here you can see that relatively compared to serum, the results that we obtain in serum, the results in milk were relatively better for our kit because the relative sensitivity was 97% compared to 93% for the competitors. And for specificity, our relative specificity was 100% compared to 97% for the ELI test kit. The second study was done here in Poland by Adrian, so you will give more details about this study in the next presentation. But here, this study aimed to compare 3 commercially available kits. So in red, this is the curve obtained with the IDVET kit. In blue, the curve obtained with the IDEX kit, and in green, the VMRD kit. And you can see, so the performance of the kits were evaluated thanks to the area under curve. And the more close to 1 is the AUC, the more performant is the kit. And we can see here that on milk, the red curve is above the 2 ones associated to the competitor. So that means that we have at least similar or even better performances than these 2 competitive tests. So what about the study that were led on sera? There is a very recent study led in 2022 comparing the sensitivity of our kit with the IDEX P28 one. And here we can see that we are very largely above the competitor because we are almost 100% and compared to a competitor which is near 80%. Last but not least, and I'm going to finish with this study, the ID screen kit was compared to all the competitors that are available on the market. So there is the ELI test, the IDEX kit, and a test kit from LSI and InFree Diagnostic. And we can see that on 50 known positive samples that were diagnosed with AGID, we obtain a sensitivity of 100%, which is not the case with other competitors. So here you have, on average, 95% of sensitivity for other test kits. So what I would like you to remember from this very quick presentation and quick overview of the literature about this ID screen kit is this kit is already worldwidely used in epidemiological studies and in population screening. This shows excellent performances with high specificity and sensitivity, and these performances are at least equivalent, if not better than other kits on milk and serum sample. And also there is good repeatability, robustness, and reproducibility. So all these data are available on the validation file. So please reach out to me and I will send you the file if you need more details. This, and Professor Kaba insisted on the need to have an easy to use and very flexible ELISA test. So this is one of the characteristics of this test. And to finish, this is a test which will not give unspecific reaction due to the vaccination against BTV8. So this is, to conclude, one very good tools to diagnose this disease and to monitor MVV or CAE infection in serum or in milk sample. Thank you for your attention, and I'm open to question if there is any. Thank you.
Zosia, do we have any questions? Okay, so there is no questions. Maybe from you,
audience?
somebody
There
from
is
the audience? Okay, yes. Giuseppe, just a moment. I will just pass the microphone to you.
Thank you very much, Adrien. It was really impressive. What I would like to know is you are talking about recombinant peptides that you are putting on the plates. Are those complete sequence or just fragment of GP135, and how they are produced in which type of system?
To be honest, the production part of your question, I'm clearly not involved in the production, so I won't be able to give you a very accurate answer on how they are produced. But this is whole antigen cocktail that is coated in the plate. This is, I'm not sure this is a recombinant one. I need to confirm that, but
okay.
I will confirm that and say it to you.
Thank you very much.
You're welcome.
Thank you very much. Any other questions? Not any other. So Adrien, thank you very much. Thank you for coming once more. Thank you, the IDVET company, for supporting us. Thank you very much. Thank you.
Thank you for the organization one more time. Thank you.
Thank you. Okay. Well, now it's time for Adrian. Adrian comes from veterinary faculty in Cluj-Napoca in Romania. Adrian stays with us already quite many years. And he did his PhD on caprolactis encephalitis. So, well, the floor is yours.
Thank you very much. Hello to everybody who are present in the room and also to those who are online. So today I will speak about the diagnostic of CAE using milk samples. We know that the antibody against small ruminant lentivirus are produced usually 1 to 3 months after the infection, but there are some cases after few more months and remain detectable for life. For this reason, it makes the serology, especially the ELISA test, the mainstay of CAE diagnostic. Nowadays, several commercial ELISA kits are present on the market and approved for scientific purposes, but only a few are registered for diagnostic use. The main difference between the ELISAs is the type of viral antigen they are based on. We have ELISAs that are based on whole virus antigen ELISA, like IDVET, recombined transmembrane encapsid protein ELISA, like IDEX, and surface glycoprotein ELISAs, like VMRD. So the blood, it's the material that is used normally to test CAE. because the blood has some good advantages. Is the most commonly tested material. All animals are included, and also we know that their accuracy has been well studied over the previous 25 years, and they appear to perform generally well, yielding highly consistent results. However, this kind of material has also disadvantages, causes stresses to animals. We can speak about the reduction in milk production temporary because of stress, of course, requires qualified personnel, legally restricted to veterinarians in many countries, which definitely will increase the cost. And also the farmers are not very happy when we need to collect blood. And they are often discouraged to participate in control programs. These impediments raise the question if the milk cannot be used as an alternative material for serum. For example, in dairy cattle, this approach has been widely applied in routine veterinary practice, for example, bulk tank milk, but in goats, the milk serology and testing is relatively rarely used. To date, only one commercial ELISA kit is approved for diagnostic use on individual milk samples, and another one is approved for research only. Which are the advantages and disadvantages of using milk? It's easy to collect, no special material are needed, no impacting on the animal welfare. This can be done directly by the farmers, the costs are reduced, and of course encourage the farmers to participate in control programs. And also we can use pooled samples. I will discuss about this a little bit later. Of course, we have also disadvantages. We can test milk only if we're speaking about dairy herd. We have also in every herd non-milking animals, for example, the young ones or the males. Not all the animals are tested, and and the animals do not have milk all year round. So to collect milk, we use usually the Falcon tubes of 15 milliliters or 50. We use to centrifugate milk because we know the antibodies are present in the liquid part. So after centrifugation, we will obtain 3 layers: the fat, the liquid part, which is called whey or lactoserum, which contain the antibodies, and the sediment. After that, we put an Eppendorf and store until testing. We know that the presence of excess fat is known to produce false positive results. For this reason, we are not testing the milk like this. So we centrifuge the milk. Why? Because the fat can influence the optical densities and so obtain the false positive results. And we can say this also from our experience. We tested also only whey and also full milk, and we had differences. So I want to show you the study of to show you the results of one study that we already published. We wanted to see if the milk can be used instead of serum, and to test also and to determine the accuracy of 3 commercial available ELISA approved veterinary diagnostic use. All of these perform well on serum. However, their accuracy has for testing milk has not been so far evaluated and compared. There are some studies that were published on individual milk samples with these 3 ELISA kits, but they tested only the agreement, not also the accuracy of these ELISA kits. So for this study, we collected per samples blood and milk from 420 goats, originating from 5 dairy goat herds from Poland. 3 were known to have no history of small ruminant lentivirus infection, and 2 were known to have CAE. For this study, we used 3 ELISA commercial kits, 2 indirect ELISA, 1 from IDVET, 1 from IDEX, and a competitive ELISA from VMRD. So the ELISA were performed according to manufacturer protocols for serum. The serum was diluted 1:20 for indirect ELISA and remained undiluted in competitive ELISA according to the manufacturer. However, the dilution for milk was not known. And here you have an illustration of how the situation can be. We know that the concentration of antibodies in milk is lower as compared to sera. So we need to check what is the dilution we need to apply for milk. So for this we selected 5 milk samples from 5 seropositive and 5 seronegative goats. We performed twofold dilutions starting from undiluted to 1 to 128. And in the end, we choose the 1 to 2 dilution for both indirect ELISA because the difference in the optical density was one of the strongest. As you can see in this picture, this is IDVET and this is IDEX. The dilution 1 to 2 was the best, and this situation was also reported in other studies. After we tested the samples, we obtained positive results in 235 goats using the whole antigen ELISA. 231 goats were positive using their recombined transmembrane capsid ELISA, and 211 was positive in VMRD ELISA. What's the situation regarding the milk? When we performed the ELISA using milk, the number of samples were similar with those obtained in serum. We checked also the area under the ROC curve for all 3 ELISAs, and as you can see in the indirect ELISAs, the area under the curve was really high, roughly 98%, and for the competitive ELISA was 92%, as you can see also in this picture. The optimal cutoff value for milk was different than the one for serum, as you can see here. All the optimal cutoff value for milk were lower than those in serum. These optimal cutoff values, both indirect ELISA, were expected to yield highly trustworthy results. Predictive positive value and negative predictive value of at least 90% for a wide range of prevalence of small ruminant lentivirus infection, from 30 to 80 in terms of whole antigen ELISA, or from 30 to 70% in the other indirect ELISA, as you can see in these images. However, in the competitive ELISA, no prevalence was able to ensure both a positive predictive value and negative predictive value of at least 90%. The agreement beyond chance between the results of each ELISA performed using serum and milk samples was very good for both indirect ELISA, however, only good for the competitive one. You can see in these tables and here the beyond chance agreement for the whole antigen ELISA was roughly 92%. For recombined ELISA was roughly 93%, but for the competitive ELISA was 76%. So what are the conclusion of our study? Our study shows that the accuracy of the indirect ELISA done on milk samples appear to be similar to their accuracy on serum samples. So we can use milk sample instead of sera. Accuracy of competitive ELISA is considerably lower. Diagnostic sensitivity and specificity of this ELISA appear not to be lower on milk than on serum if we use, of course, the proper dilution. I want to say a few words about the bulk tank milk testing. This approach is currently used in dairy cattle for screening herds for various infection and parasitic diseases, including diseases from the OEL list. In goats, serological bulk tank milk testing was so far evaluated in different infection and parasitologic diseases, including small ruminant lentivirus infection. So what are the advantages of using bulk tank milk? All the advantages that are for milk, of course, but also time saving to collect. And we are speaking about only one sample, which is representative for all milking animals. However, the bulk tank milk samples has also disadvantages. We don't know the proportion of positive animals we need to have to obtain a positive result. The volume of milk the goats yield is variable, and also the concentration. We can have goat with high titer of antibodies and goat with a lower titer of antibodies, and this can influence the bulk tank milk results, of course. So these disadvantages might lead to false negative results. So it should be noted that a single collection of bulk tank milk may not be enough to determine the true status of CRLV infection, especially in the herds where we have a low within herd seroprevalence. Such problem can be overcome through repeated measures. An author suggested a serial sampling strategy, testing bulk tank milk samples several times each year in order to include all lactating goats in at least one bulk tank milk sample. And it is very likely that herds with a low within herd prevalence will test positive a later sampling. We made a small study in Romania. We investigate 25 herds from Transylvania using bulk tank milk testing, and we found out that 20 of them were positive. Of course, we didn't test also blood in these herds, only in a few, but in those which we tested, the results were in agreement with the bulk tank milk testing. And I want to end with one more slide and tell a new concept that we developed this year. We published a study using equal volume milk testing. What does it mean this? This is a combination between the collection of individual milk samples and the bulk tank milk. This means that we need to collect individual milk samples from goats, and in laboratory we will take the same volume from this sample, and we will create an artificial bulk tank milk sample or an equal volume milk sample. So you can see here the difference between them. Here we can control the volume that is used. So here is the study. I will not I will jump directly to the conclusion because we don't have time to say more. But only two words about how we did it. We selected 100 negative milk samples from negative goats and 100 positive samples from positive goats, and after that we create artificial prevalences from 1 to 100 by diluting positive milk samples in negative milk samples. So our conclusion are that we obtain a very strong monotonic relationship between the proportion of individual milk samples from goat seropositive used for preparation of these samples and the optical density value measured using commercial indirect ELISAs. This kind of testing overcomes one crucial weakness of bulk tank milk. Inequality of milk samples contribute by individual lactating females. However, because this is a laboratory experiment study, we have also some drawbacks. So we need to check also if this will work in the normal life, because this was experimental study. I want to thank to the organizer and those who helped me, especially to Jaroslav Kaba, Michael Chobovic, to all the team from Poland and from Cluj, and thank you to all the organizers. Thank you very much.
Thank you very much, Adrian. Zosia, no question from the world. What about the audience here? Is any question here? Not any question. Okay, not any question. So we are really very fast. Thank you once more, Adrian, for coming and for presenting. And well, we are also happy that you are staying with us, and I hope for a longer time also. So thank you very much. And
Thank you.
Martin, welcome. I think everybody knows Martin from Hanover.
Why?
Because you are the important guy in goat diseases, also in Capran arthritis. So thank you very much that you come, and well, welcome. So the floor is yours.
Thank you for inviting me, despite I'm not part of this group here, and maybe this has more historical reason, I think. So the talk should be on, but you will see it's a little bit deviating from this theme. For we have a long history already on CAE sanitation, and it began already in the 90s. And till then, the number of goats increased. We had in the 90s about 80,000, 90,000 goats. Now here is a census of 2016. Now we might have 170,000 goats, which is not too many, but it's increasing. And especially the milk production is increasing, mainly focused in the southern federal states, Bavaria and Baden-Württemberg. But the main problem is that we have a lot of very small farms with less than 50 goats, and even a lot of hobby farms which are not really interested in production or so, and it makes no sense to focus on those. But we have to focus on these milk producing herds. They have about 30, in 2016, they had about 35,000 goats. I think now it's much more than 50,000. At that time, the average goat herd of these producing herds had 116 goats. Only 11 farms had more than 500 goats, and most of them, 6, had not only goats but also cows. And the number of these bigger farms increase continuously now. So there is a need for the control to change. There's a pressure from the market. And what we have to see, all these bigger farms are sanitated, or they were built up from sanitated farms. So the prevalence of CAE in these big farms is nearly zero. You sometimes have some individual problems, okay, but altogether this CAE is not really neither a clinical problem nor a problem of diagnostic. A more difficult thing, special thing in Germany is that 2/3 of these producing farms, are working organic, which is a difference to most of other countries. If I look at the numbers of tests we do in my lab, which is local at the university, we're doing mostly the tests in Lower Saxony, but we have also samples from other federal states. Concerning the number, it was relatively stable from 2001 till 2021. What changed is the number of positives. In 2001, we still had 8% CAE positive sera. In 2022, we had, and 21 also, we had less than 2% CAE positive. And we are a clinic, so we also have those hobby animals where the owners don't care about this, but we diagnose it, CAE in those animals. So that is the good thing in the change. The bad thing is that the number of samples and also farms who are sending in is declining, especially in the last years. The costs are increasing, and I will show you why the farmers complain, especially of these big farms. The structure of the dairy goat industry, even it's organic, it's changing. The farms and the herds become bigger. And we have a structure. You see, you have some areas like here, for example, where some of in the areas there are more than 1,000 milking goats. Concentration here in Bavaria, then here in Saxony and Thuringia. There are increasing numbers now here in the West, which is due to the fact that the milk they produce, it's going to the Netherlands. And nowadays here is a new focus. It's not at this slide, for there is now a very big dairy, organic dairy, producing a lot of milk powder for human consumption, which is an interesting market. So there is a change, especially to the big farms. The control program focus on the Swiss program. It's adapted to that. So if you have no status on CAE or MADI, it's the same for milking sheep herds. The first thing is it has to be a closed herd. The first test of all goats is in all animals which are older than 12 months. If all these tests of the whole flock is negative, 6 months later there is a second serological test of all animals. If it's negative, a third test with negative, and after that the fourth test is, if all these tests are negative, the herd is regarded as CAE or MADI negative. And all these tests have to be repeated every year. So imagine if you have 500 animals, milking goats in such a herd, you have to test every year 500 animals, which are enormous costs. The problem in Germany is that federal structure. So in some areas, some states, these testing or even sampling is subsidized by the state. In other areas, you get nothing from the state. So there are big differences. For example, in Bavaria, it's subsidized. In Lower Saxony, nothing. Okay. There are also some hygiene restrictions, and especially injections shall be carried out with separate disposable needles for each animal. That's very important, especially if you make vaccinations or deworming. We have now this decline in the sending in of the samples from the farms, especially from the bigger farms. They asked, isn't it possible to reduce it? Or they say, I'm not a breeder. I see no more effect. I'm now more than 10 years negative. It's too high. The costs are too high. So we have to discuss how can we control these farms, but more cost effective, and also sure enough to ensure that they're still negative. And if we do that, we have to look to the Netherlands, and probably it would be better than some of the Dutch guys, René van der Brom or so, would stand here to explain this system. They also test, it's a lot of articles in their control program, but I want to focus only on that, which is regarding the control of negative herds. So again, they have to be tested every 12 months. It's somewhere written here. Yeah. And what they don't test all individual, but they test only a part of them. And you see here, they focus on a prevalence to test the prevalence or find the prevalence of 2% with a confidence of 95%. And that means even if you have a herd of 10,000 goats, you don't have to take more than 150 samples, which is relatively low. And I knew there is something, they are doing something with bulk tank milks and so on. And so I wrote to René, how do you do it? And he answered by email, we test dairy sheep and dairy goat farms in bulk tank milk samples for CAE and MEDI by ELISA. This is not officially in the control program, but additionally. And on farms which are accredited as free for long years, they pool the samples from the sample size in the lab and test them in pools of 5. In case of positive pools, they test the samples individually. So this bases on relatively old investigations, mainly from Van Manen, and they could show that the sample, naturally, you have a sample dilution if you increase the pool size. So from 1 or 2 per pool to 10 per pool and so on. So the sensitivity goes down very quickly. And they showed that especially in goats, the relative sensitivity is declining very quickly. If you have, for example, a pool of 8, you only have a relative sensitivity of 50%. And to summarize, fractions of low titers does hardly allow pooling, especially for goats. And if you pool 2, 4, or 3 correspond with a loss of sensitivity of 4, 29, or even 50%. So that sounds not very good altogether. There's another question, the pooling. You have these technical problems, but also economical problems. And the result and cost-benefit effect of pooling depends mainly on the prevalence. If you have an extremely low prevalence, it's cost-effective. But if you focus on the prevalence of, let's say, 2%, a pool of less than or more than 5 samples is not effective anymore. So polling is only feasible for monitoring of certified herds. And in the lab, you have to have a good logistics. You need the sample stream on one side for the monitoring, for all these pooling, for you have to freeze the individual samples on the other hand, and you have to divide it from the other testing where you have positive herds, or you test new animals which are brought into the farm. So that's individual testing at all. That's the difference. So they summarized in that a pool of 1 to 5 is economically and technically feasible for monitoring of certified herds. It reduces the costs by 40 to 50%. You have to reduce the cutoff of your ELISA. They used the inner test ELISA, which is not mentioned up to now. It's a relatively expensive ELISA. That's a problem for me. You have to sell it when you have a relative sensitivity, and you lose the borderline sera by this. Okay, this is bad news, but if you repeat it every year in such big flocks, it could be effective. So they also used the testing of milk samples by antibody ELISA, and you see here the difference in, as we heard already, the ELISAs are relatively good. We have a sensitivity of 98%, and which is really high. It's a little bit lower in sheep, but that doesn't matter. So you also could use it in milk, these tests. And if you use bulk tank milks, you have even you can use also the PCR, which is done now in the Netherlands, which is new. They used two different PCRs, one focusing on the GAC antigen and on long terminal repeat. And you have, even in bulk tank milk, agreement of 90%. What we have to know that they have in the Netherlands a system where every month bulk tank milk from these big herds are taken and sent to the lab due to Q fever. They control by this Q fever by PCR, and that's the reason why they had the idea to use these samples also for the PCR, for CAE. So we will discuss with this autumn in Germany, together with the other health services of the federal states, how we can equalize our testing in the different federal states. So still we have the possibility to test all the animals. And I think that even in Saxony, they will remain on testing that, for they have the money. They are subsidized. Okay, do it. No problem. The farmers are happy with that, but we have to reduce. We could, for example, reduce the testing by only taking 10% of the goats and test them by ELISA, either with serum or milk. I think we probably will use that system that we take a risk-based random sampling of individual sera, test by ELISA. This is easy to establish in all the labs and in all the health service. I think that all the other health service, they say, oh, our labs don't like pooling, so they avoid it. What we did in our health service in the last year, we took all the animals, even in these big herds, and tested that by pools. That meant sera. We reduced, let's say, at 500 animals, these 500 samples to 100 pools, which reduces the costs very, very much. But as I discussed it with the other health service, they refused it. This is nothing for us. They don't want to have it. So probably we will remain. But maybe we can discuss this pooling if we can establish a routine bulk tank milk sampling of these big herds and test it by PCR. That's a proposal and thinking for the future. I don't know how my colleagues in the other health service will decide. We will have a consensus meeting in autumn, and I hope we really find a solution. Thank you very much.
Thank you very much, Martin. Well, you said that you don't have the connection with the CAE. Everybody can see that it's not true. And well, I am also happy that you are not the first time in Poland, not the first time on our faculty, many times already.
Already, yeah, and since a long time.
Yes, exactly. So it's also a great pleasure for me that you came also this time. Thank you very much once more.
Thank
Zosia,
you.
do we have any question? No question. Okay, maybe somebody from the audience.
I hoped.
So when you do all this testing and you get a positive in a big herd of 1,000, what's happened there?
first of all, we test whether it's not a false positive. Okay, so we make all the other, we send the sample to the reference lab. If they say, okay, it's positive in more than 2 tests or minimum 2 tests, this animal will be killed and we will try to find the virus by PCR or culture. If that's found, it's necessary to repeat from the very beginning, testing all the animals. No chance at the moment. If the other, it's a decision of the farmer. If he says, oh, that's much too expensive now, we go out of the system. But that will be a very critical situation in the future. And we had it already. They remained in the system up to now, but you never know.
Thank you very much.
So thank you.
Thank you once more. Thanks a lot. Bye-bye. Martin is leaving, so have a safe way back home. Thanks a lot. And, well, the next presentation, it's Professor Emilia Bagnicka. Emilka is, well, we know each other for 30 years, better don't mention. Emilka is from the Institute of Genetics. I think the name is another, some very complicated.
Genetics, Animal Biotechnology.
Oh, something like that.
Polish Academy of Sciences.
Yeah, that's the most important, Polish Academy of Science. And well, we work together with Emilka in this subject already many years. We used to have the herd, infected herd in the institute, and we did a lot of studies on those goats. But unfortunately, this is the end. There are no goats in the Institute of Animals Genetic. But still, Emilka has a huge experience with capsular arthritis encephalitis.
Yes, but quite another side, because I interested in the genetic background of the immune response of the organism of goats. But we cooperated in many projects, and I hope that it will be still.
Yes, let's hope. I'm quite sure. So,
thank you.
The floor is yours.
So Professor Kaba asked me to say some practical word about experience in rearing CAE-free kids, and let's move to the information. And at the beginning, some words of introduction. As you know, the virus attacks selected cells of the immune system, and it is long-term and slow inflammatory changes in various organs in animals. And as Professor Kaba said, it leads to painful lameness, loss of condition, and emaciation of the goats over time. And Professor Kaba said about symptoms, and I will not repeat this information. From the breeder point of view, the most important information is which ways are of spreading of this disease, and there are two very important ways. First one is vertical, and the main way is from the infected mother to the offspring through colostrum and milk, and the second one is horizontal through long-term direct contact between animals. But there are, of course, more risk factors of spreading of the virus, and there are milking machines, needles, tattoo, surgical equipment, and also seropositive bugs during mating. I will say about it more. And some researchers indicated that it is possible to infect it during pregnancy, but it is a very low probability. And some researchers found the virus in drinking water and air exhaled from the infected animals, but this way wasn't proved that it is possible to infect it in such way. In Poland, unfortunately, there is no eradication program, and this information are from Professor Kaba, who started with this to study this disease so many years ago. It was the end of the last century, and when he started his study, only 26% of investigated herd had infected animals, and among these herds, 6 owners imported their animals from abroad, and if I remember well, they imported these animals from Western countries, and among these 3 from this disease herds, only one owner imported his goats from abroad, and if I remember well, it was from Ukraine. Yes. So the next study Professor Kaba and his team conducted in 2002, and after several years, 40% of investigated herds had infected animals. The next study in 2007, and it turned out that almost 70% of herd had infected goats, and these results were confirmed for the next study conducted for several years. So we can say that more than 60% of herds in Poland are infected with this virus. Of course, there is no possible to cure the animals, and infection is eternal, so to the end of life of goats. But we can try to eradicate the virus from the herd or from population. And of course, the basic thing is the testing of animals in all herds, and then the one possibility is to eliminate of all infected animals from herd or from population, but of course, it is a very expensive method, and without supporting of governmental supporting is impossible. And this happened many years ago in Iceland, and this island is free from this virus. According to knowledge from Professor Kaba, there are a lot of herds in Poland where almost all goats are infected. So this procedure should be very expensive and is rather impossible. So, but there is another way for eradication of the virus from the herd. We should also test all animals, and if part of animals is seronegative, we should divide our herd into 2 groups, and of course these animals should be isolated from each other, and all newborn kids should be isolated from their mother immediately after birth. And the best way if the goats even doesn't lick their kids. And then kids are reared on cow colostrum, on cow milk, or cow replacer. Some researchers say that it is possible to use goat colostrum, but this goat colostrum should be pasteurized because the virus can be present in milk. And necessary requirements in this procedure is to test systematically all animals, and infected animals must be gradually eliminated from the herd. And as I said, no official eradication program is in Poland, but such program was introduced in several herds in Poland, and I would like to say you some information about the results. This program lasts usually for several years, but so 3-4 years could be efficient, but of course it is very labor intensive and very expensive program. And effectiveness depends on the determination of the herd owner, and especially during kidding time. The proper organization of kid rearing and maintaining healthy animals isolated from infected one. And each breeder should be aware that the program can fail due to improper supervision during kidding and this lack of isolation. When I prepared this presentation, I found in internet that now it's possible to buy some special bra for goats, and I think this bra could be very useful and help breeders to supervise the goats during kidding to avoid drinking milk by colostrum by kids. So if breeder think about eradication of the virus from the herd, he has to think about prophylaxis. And first of all new animals should be serologically tested before being introduced to the herd. And as I said before, the serological status of bugs should be also known, because during breeding, the male comes to direct contact with many females, especially with harem mating. So mating season lasts sometimes 2-3 months, so it is enough time to introduce the virus to the herd. And of course, in large herd, the owners could use their own bugs, but of course, he should monitor the inbreeding rate. But when breeders think about the eradication of the virus using this program, the question arises: if the offspring is offspring properly reared when we weaned them from their mother just after delivery. Therefore, we analyzed our results and we wanted to determine the differences in the rearing results of kids with their mothers and those weaned immediately after birth and fed with cow colostrum and cow milk. So some words about method. This program was conducted in herd, consisted of approximately 50 goats. The goats were in good physical conditions. Many goats had no symptoms of CAE. Some had mild symptoms. For the 3 years we conducted this procedure, and during first and second year all kids were weaned just after kidding. But in third year, kids from seronegative goats were with their mothers for 3 weeks, as usually was done in this herd area. And we built 2 groups, and experimental group consisted of almost 200 kids, which were weaned just after parturition. And the control group were kids weaned 3 weeks after birth. And this group consisted of these kids born in the third year of the study, and of course 40 kids is too less number to conduct any analysis. So we used data on more than 400 kids born in 4 previous year in this herd, and it was routine to left kids for 3 weeks with mothers. So the first period was colostrum period. It lasted 5 days, and kids were fed 4 times a day with colostrum. Fortunately, we had a dairy cow herd in the next village, and before kidding season, we collected a lot of liter of colostrum. We collected only colostrum just after parturition of cows, so this colostrum was full for proteins, and we stored this colostrum at -20. Of course, the colostrum was melted. Then we diluted the colostrum with water because, as you know, the cow colostrum is usually very thick. And then this colostrum was heated up to temperature of 37, 38. As you can see in the table, there is the doses of colostrum, and in 5 days, it was almost 1 liter colostrum per day. Then the animals obtained fresh cow milk. Of course, milk was also heated up to 38, and they drank 3 times a day up to 12 weeks of life. And the maximum dose was 2 liters of milk per day per kid, and it was around 5th week of experiment. And after the second week of life, the kids had access to the good quality hay, and after 3rd week of experiment, they obtained also crushed oat. And summarizing this, the economical side of this rearing, this way of rearing of goats, we used 120 kilo of cow milk for 1 female kid and 150 kilo milk for a male kid. And we used also the concentrates. It was between 150 and 300 grams per day, but depending on age, of course, and hay was ad libitum. And it is also worth to mention that kids weaned after 2 weeks were fed the same as peers, so they also obtained cow milk and concentrates and hay. Of course, all animals, all goats were tested in November of each year, and all born kids were tested at 3 months of age. And some part of kids were for renovation. It was and they were in the herd. And they were tested several times after this period. Of course, we used serum for this analysis and ELISA test. And here is written the name of this special test of ELISA. And we studied body weight at birth, at the age of 7 days, at the age of 11 weeks, and daily gain from birth up to 1 week and up to 11th week. And we conducted statistical analysis and using mixed procedure. And we used model with the random effect of the mother and the fixed effects of rearing system, season of birth, number of kids in the litter, kids sex. And also we used fixed regression of the birth weight of the kid. And now there are results. At the beginning of the experiment, almost 70% of adult goats were infected in this herd. And as you can see, in each year, only one kid was seropositive at 3 months of age. And probably these 3 kids drank colostrum from the infected mother just after parturition. Some part of kids were tested later, and all of them were seronegative. And now I would like to show you the results of the rearing. And the main parameter is the deaths, the rate of the deaths of kids. And as you can see, the percentage of died kids are similar between rearing system, and of course it depends on the year. And usually it wasn't the influence of the rearing system, but rather it was influence of the staff which work with goats. And regarding the traits of the body weight and daily gain, as you can see, there were no differences between body weight and in 1 week and in 11 week, and daily gain up to 1 week. And we found only differences in daily gain from birth up to 11 week, but as you can see, there is no big differences. And now I would like to show you the chart which present the information about prevalence of the virus in this herd till the start of the program of eradication. Each year, almost 8% of goats, so the number of infected goats increase in each year. And after implemented this program of eradication, the number of infected animals dropped almost 10% each year. And after, I think it was after 4 or 5 years, only 25% of goats were infected. And in this moment, the owner should eliminate all these animals from the herd, but it doesn't happen. But it is quite another story why. So, and as you can see, the number of these infected animals was similar, so what on the same level in next years. So as a conclusion, we can say that although the eradication program is lengthy, laborious, and expensive, the virus can be effectively controlled in the herd. And so having healthy goats seems worth to effort, but effort is really big. Thank you for your attention.
Thank you very much, Emilka. And well, I would just only mention that it was not so easy to rear those kids. The students from our faculty, they have been helping us a lot, you know, because that was really a work.
Yes.
They stayed over the night, you know, and 24 hours a day. So it was not easy, really not easy. But well,
Yes.
As you see.
But as you saw, now there is the bra for animals. We haven't it, and I regret it very much.
Yeah.
So now would be easier, really.
Yeah, but the other thing, what you mentioned already, it's you know, the owner. The owner really wants to do it because, well, we put a lot of work, as you have seen on this chart, and then there was no decision. So yeah, the whole work is, well, for nothing, in fact. So first of all, in my case, if we have to deal in the goat herd, so we speak with the owner, and he or she has to decide and really decide, because and then keep this decision. Otherwise, it's useless. It has no any sense.
So
Do we have? Sorry. Yeah.
Unfortunately, there is no support from our government.
Yeah, of course. That's the most important thing. No support from the government, from any organization, so they have to pay for themselves. Do we have any questions?
Oh, it's about Jones disease when feeding cow colostrum and milk, and I think it's a question to you. It's not for me. It's about Jones disease.
Well, but this is, well, I would not like to discuss this at the moment, because the whole meeting is about capran arthritis and encephalitis. So let's leave the Jones disease on the side. And openly speaking, well, we ourselves, we don't have great experience with Jones disease, because thanks we don't have a lot of cases. In fact, not so many cases in Poland. We are very happy of it. That's another story. Well, Emilka just showed you how the capran arthritis was spreading in Poland. It was spreading just because we were buying the infected animals, infected goats to Poland. With this Jones disease, we face now the problem, because we see that the prevalence of Jones disease in goat population in Poland is increasing. For many years, it was just really very few cases, but now it starts increasing. And well, the problem is that well, the animals, infected animals, they are coming to Poland, because in Germany they are trying to eradicate the Jones disease. And you know how it is. So and those infected animals, the price of those infected animals is low, and our breeders don't care. So I'm really afraid that you have seen how it was growing, the prevalence of the disease of the capran arthritis in Poland. I'm really afraid that in 5 years we will organize the same conference about the Jones disease in Poland, and showing the similar numbers, and then maybe will be the time just to discuss the Jones disease. Let's leave it just on the side today. Do you have from the audience any other question? Not any other questions. So Emilka, thank you very much. Thanks a lot. Thank you very much. And Saulius, please come to the stage. Saulius will show us the study on capran arthritis and encephalitis, which was done in Lithuania. Well, I think, Saulius, it's the first such study in Lithuania. Am I right?
I'm sorry for sitting. And a similar situation to Poland, and we importing some goats and sheep. And let me start my presentation. And in Lithuania, it's not like a small ruminant country. It's a tradition that is large ruminants, pigs and poultry. But particularly situation changed something about 15 years ago, when appeared in Lithuania African swine fever. So the situation, it was, tradition was a little bit changed, and small ruminants population started to increase significantly. And majority of them, they've been imported from somewhere, and nobody knows what they're carrying really on. And it was from Germany, from France, from different countries. And particularly increased sheep population was few times. Goat population still not. I was wondering always that Lithuanian goat population is lowest in the EU, but it seems that in Sweden is low amount of goats. They just have 6,000 of goats. We have something like about 9,000 of goats. So, and there is no mandatory testing of caprine arthritis encephalitis in Lithuania, and farmers can order testing only individually, and many farmers do not know this disease and it's causing problems. So what we decided, we just printed, even because maybe they're not looking somebody to internet so much. So we printed information, one kind of disease when we started this. So, and performed research about this virus situation in country. And we share this information, and I will tell you what we've done at the moment. So we selected goats from various regions in Lithuania, and we start to take blood samples and screened them with a commercial ELISA kit, which is company which supporting this meeting. And farmers agreed to participate in the study like volunteers, and we have visited herds where at least is 10 goats in the farm, and we collected 380 blood samples and tested with ELISA from 30 goats farms. So it's not a low amount if you take amount of goats in Lithuania. So we are a small country, 65 square thousand kilometers. So we taken samples from, you see, all countries, all country around Lithuania. And together with sampling, we made special survey for farmers in order to determine health and productivity problems. And it could be done by this virus in goat farms. And it was around 30 goat keepers interviewed. And we found that its main noticeable symptoms of joint damage and respiratory system disorders are in goats. And only one farmer, he talked about neurological symptoms. And in the next stage, the survey data will be compared to the obtain of laboratory test results. So some pictures we made, and it's also is here some which is made for with like in Lithuania. So like it looks joint damage in seropositive goats. Also, we done in Poland some PCR testing. Thank you for Polish colleagues, and 10 of 17 seropositive hairs from various Lithuanian legions were tested by PCR by this reverse transcription nested PCR, and those 10 hairs which it was positive were revisited, blood was collected, and only from seropositive goats, and we took to Poland these some samples, and we checked by PCR. I will tell later about this result. So if you take with ELISA from 38 blood samples, it was 74 positive, quite a lot. It's close to 20%. And in 17, more than half farms of 30, at least one seropositive animal was found. And 22 of 27 seropositive goats were positive when we checked with this reverse transcription nested PCR. And goats came from 9 of 10 tested herds. It was around 90%. So both this small ruminant lentavirus were identified, and 8 samples they were like medivirna virus. It's more common in sheep, of course, but we found also in goats. And 14 sample, it was like a arthritis encephalitis virus. And so conclusions, it's not very big study, so it's more than 56% of goats farms where they had at least one seropositive animal, and both small rumen and lentavirus genotypes and be are widespread in Lithuania goat population. So what in the future we're going to do, so we planning to do genotyping, and we will compare results of survey with laboratory results, and we will try a new test. So shortly, that's it I want to do.
Thank you for updating us with the situation of capronephritis encephalitis in Lithuania. Any questions, Zosia? Not any questions from internet, from the audience? Not any questions? Thank you very much. Thank you for coming, and thank you for presenting the results. And the next presentation will be about the situation of capronephritis encephalitis in Hungary. Laszlo, please let us just introduce us with the results of your investigation, which was done in Hungary between the same project, the ICRAT project. Thank you. Thank you very much for coming, and the floor is yours.
Thank you very much, dear colleagues, dear ladies and gentlemen. I came from the University of Veterinary Medicine in Budapest, and it was the first national survey of caprine arthritis and encephalitis in Hungary, and I would like to show the results of this survey to you. Just a short introduction about the Hungarian goat sector. Frankly speaking, this is a quite marginal sector amongst the animal keeping sectors, but the Hungarian goat population in the last 3 years at least slightly increases. There are both meat and dairy goat herds within the country, and goat products, dairy products, especially the different goat cheese products are getting more and more popular, especially the homemade cheese products. We have very few large dairy goat herds and very few, less than 5 goat milk processing plants, commercial goat milk processing plants. There is a new trend that the agrotourism, the pet goats, keeping pet goats, I mean the hobby goats, are more and more popular. The little children, the kids, like the goats very much, and that's why, for example, in Budapest and in the suburbs, the goat population is increasing. About the animal health status, the status of the different infectious diseases in the goat herds, we have very little information about the infectious diseases, the health status of the goat herds, and so far we have no national prevalence data about the caprine arthritis and encephalitis virus. So this graph shows the number of the female goats older than 1 year in Hungary between 2008 and the last year. And as you can see, there is an increase. There was an increase till 2018. After that, a slight decrease, and in the last 3 years, a slight increase can be observed again. And we have around 25, a little bit more than 25,000 female goats and their progeny, of course, in the country. So this is not a large number. This data comes from the Hungarian Sheep and Goat Association. This graph is about the number of goat herds in Hungary. So as you can see, between 2008 and 2022, the same period of time, and we can say we have a little bit more than 1,000 goat herds in the country, between 1,000 and 1,100. So the number is varying between these two numbers. And this table shows us the distribution of the herds and the female goats according to herd size. And as you can see, most of our goat herds are very, very small, very small. Between less than 10 goats were kept in around 1/3 of all Hungarian goat herds, 31.2%. Between 10 and 20, another third, a little bit less than another third. As you can see, the number of registered goat herds out of the total. And we have very few large herds, over 200, female goats can be found only in 4 herds, in the entire country. And this graph shows the distribution of the herds and female goats according to the herd size. And you can see the blue bar showing us the share of the goat herds in percentage out of the total, and the brown orangish bar showing us the share of the female goats, the number of the female goats. And as you can see, despite the fact that most of our herds, 2/3 of our goat herds are very small, they are keeping less than 20 female goats. Around 40% of the entire goat population are kept in the larger farms with more than 50 female goats older than 1 year. Just a few words about the breeds which are kept in Hungary, the goat breeds which are kept in Hungary. Dairy goat breeds, the most popular one amongst the dairy goat breeds, the Alpine, the Saanen, and the Anglo-Nubian. For meat goat breeds, the boar is the most popular one. And we have a so-called traditional goat breed. In English, it sounds quite strange. I mean its name, the Hungarian wasteland goat breed. In Hungarian sounds better, parlagi. So this is a multi-purpose goat breed, basically. So this is the traditional one. And as you can see, in the largest number, the Alpine is kept in Hungary. The second most popular is the Hungarian wasteland. Then comes the Saanen, the Anglo-Nubian, and the boar. The prevalence study. Our aim was to estimate the true prevalence of CAE virus in the Hungarian goat herds, and not just to survey the proportion of truly infected herds within all herds. It's so-called the herd-level true prevalence, but the proportion of infected animals within the infected herds. This is the conditional within herd animal level prevalence. So the CWHP, that was the other parameter which we were looking for. We collected the samples between June 2022 and February 2023. We collected samples from 53 randomly selected dairy or meat goat herds or both from all the Hungarian statistical regions. Hungary is a small country. We have only 7 regions. We also used a questionnaire. A farm questionnaire was filled via personal interview. So we visited the herd and we asked the farmers, farm managers or the farm owners, and we filled the questionnaire together. Altogether, 1218 blood samples from identifiable goats older than 1 year were collected. We took the samples, the blood from the jugular vein, and the sera were serological tested. All the sera were serological tested by the ID screen indirect ELISA in the laboratory of the University of Veterinary Medicine, Budapest. Just some photos from the surveyed dairy goat farms. This was a larger one. They kept more than 100 goats older than 1 year, 119 at the time of the survey. You can see this is the milking group and other younger animals. This was the milking house, another younger female groups. Yes, another photo from the milking house as well, and another production group. Just an example for the field survey. On the left side of this slide, of course, it's in Hungarian. We tried to Polish, but it didn't work out. So we translated it to Hungarian. As you can see, this is the identification number for the farm. And here are the samples, the blood samples taking, and this is the identification number as well. the farm identification number, and the accompanying documents with the samples. And this was the goat sample sheet what we used. This is the empty one, and this is the filled one. As you can see, this was the number of the tubes, the identification number of the goats which were sampled, the sex, female, male, and the clinical signs, if any, swollen joints, abscesses. These were the facts which were registered. And after that, we did the statistical analysis based on the serological, the results of the serological test. We estimate the herd level true prevalence and the conditional within herd animal level prevalence if the herd was infected with the virus, how many animals were infected individually. And we investigate the effects of herd size and regions on the prevalence of the virus. We set up 4 herd size categories: very small herds, less than 20 goats older than 12 months, the small herds between 21 and 50, medium-sized herds between 51 and 100, and large goat herds. More than 100 goats were kept in those herds older than 12 months. We used the Bayesian statistical model, which included the herds as a random cluster, and we used the RSTAN and the AirJax and RunJax statistical packages to do the statistical analysis. This slide shows the geographic distribution of the surveyed herds and the number of goats sampled within this herd. This is Hungary, and you can see with different colors the 7 regions. This is Budapest and the central regions. Slovakia is here, Austria, Slovenia, Croatia, Serbia, Romania, and Ukraine are our neighboring countries. And as you can see on the larger graph, the number of the sampled herds, yes, the table shows this, and the number of the sampled goats within the regions. Altogether, 12-18 sampled. Altogether, we sampled 12-18 goats older than 1 year among 2,404 eligible goats from the 53 surveyed goat farms. And you can see the distribution of the surveyed farms according to herd size by number. Of course, we sampled in the largest number the very small goat herds. They kept less than 20 goats older than 12 months. And you can see the other categories. The number of the sampled herds varied between 6 to 11. The median herd size was 19. The median herd size was 19. This table shows the apparent prevalence by herd size category in the sampled goat population. And you can see the number of the tests, the negative, out of all the tests done, the negative and the positive test results, and the apparent prevalence according to a herd size. And this is the final number. As you can see, the apparent prevalence on animal level was a little bit more than 31%. So it means that out of the 1,218 samples, 373 proved to be positive. And the herd level, the apparent herd level prevalence was 41%, more than 41%. It means that out of the 53 goat herds, 28 was positive. 28 was positive. And after that, we made the Bayesian statistical analysis. And I would like to emphasize this row. This is the most important one. This shows us the weighted mean herd true prevalence, HTP. So this is a predictive value, of course. 29% of the Hungarian goat herds, at least in this sample, is positive, is infected by the virus, 21%. And you can see this is the credible interval, so 95% credible interval, which means that the 95% of the goat farms have a prevalence value between 20.8 and 38.5% positivity. So these are the, we can say these are the minimum and maximum values. But this is the mean value. And on animal level, if the herd was infected, according to the statistical analysis, 58% of the animals within the infected herd was positive or can be expected to be positive, and these are the minimum and maximum values within a 95% credible interval. So summarizing our results, the herd true prevalence is 29.1% in the Hungarian goat herds. The animal level prevalence 58% on an average in the infected herds. All results showed that there was no substantial effect of the regions on the herd true prevalence and the animal level prevalence of the infection. And regarding the herd true prevalence, we can say that we can divide the Hungarian goat herds into two major groups, the very small and the small herds. So the goats, the number of the goats are less than 50, the goats older than 12 months, and the medium and large herds, more than 50 goats. Because in terms of the infection, because for in the very small and small herds, the infection rate of the is less than quarter, 25%, but amongst the medium and large herds, the infection rate, the herd level infection rate was more than 75%. So we can conclude the larger the goat herds, the infection is more prevalent. And this is an extra slide. This is not about goats, this is about sheep. Traditional sheep breeds in Hungary. We have three so-called ancient or traditional sheep breeds, the Cikta, the black-headed Cigaya, and the Racka in black and white. Thank you for your attention.
Laszlo, thank you very much. Well, you said that you find the highest seroprevalence in the big herds. It's exactly the same in Poland. Well, it's simply because they are buying some animals from outside, importing the animals. It's exactly the same. So, for example, if somebody would like to establish a herd of goats in Poland and ask me where to find the goats, where to buy the goats, so I always say, if you would like to buy a really healthy goats, just buy them from the really small herds, because if you buy from a big one, the probability that you will have the problem with CAE it's really high. So it's exactly the same, and you have the prevalence now not as high as in Poland, so you are on the way. Just follow us. Thank you very much. Follow us. If you don't do anything in the country, as it was in case of Poland, so it happened like it happened.
Yes, we have no obligatory regulations, provisions for this, so we can expect that the prevalence rate is going to be increasing in the future.
Yes, exactly the same in Poland. Nobody would like to put the money for the any eradication program, so it is like it is. Do we have any questions? What about audience? Giuseppe?
Not yet. Not yet, but it's yeah.
Sorry, I repeat just for the audience, but it's just about the breed because you have a very solid statistical analysis. Would have been interesting to see the breed.
We are planning to do it in the future because it's a very interesting question. Yeah.
Thank you very much.
Thank you.
Okay, thank you very much. Thanks a lot. And well, our next speaker, Snorre from Norway. He's a happy guy because you don't have a capronitritis anymore in Norway. You eradicated it. However, well, maybe you have. Yes, exactly. Please take the microphone. Well, so we are really waiting for your presentation. And once more, thank you very much that you came. And just let us know how you managed to get rid of the disease. You are very interesting. Thank you.
Okay, thank you, Jörg. We'll see. Yes, so the question is, how did we eradicate CAE in Norway? So this is the distribution of dairy goats in Norway. You see the map of Norway. So it's focused on mainly in 3 areas, two in southern Norway and 1 in northern Norway. And we have around 35,000 dairy goats. But we also have fiber and meat goats and hobby goats, but they are more distributed all over. So the number of dairy goats in Norway has stayed rather constant during the last years, as you can see here. And the average herd size is around 130. So what about the small ruminant lentivirus? Our first experience was with the Madivisna. We imported that in Norway in the 60s by the Texel breed. And it was notified in 72. And you can see on this map, because it was mandatory, and you can see here there's a spread of this infection, or at least the top here, in the 70s. And we thought we have eradicated that. But then another top here in 90s, and a more higher top in 2002 and 3. And again, we thought we have eradicated that, but then it's popped up again. And this is from the same area as the last these diseased animal. So it's a long, could be a long incubation period, or that it show any seroprevalence. But I could also say that these cases, the first cases was detected at post-mortem, at slaughter. And then they investigated the herd and the surrounding herds, and they found nearly 50 flocks were found infected. So here you see the years here, and you see the number of flocks, the sheep flocks, and you see the flocks that were analyzed. And perhaps the most interesting thing is this column here. You can see the number of positive flocks, and then you have this positive flock that occurred again. So that was a short about the MADI situation. And what about CAE? The antibodies was detected in 1975, but nothing was done in order to prevent the infection. So there was another study that was published in 98 indicating that 86% of the goat herds were positive. So that was a dramatic situation. So then they started an eradication program in 2001. That was ruled by the industry, the dairy organization TINE, and also from the Norwegian Goat Health Services. It was called Friskere geiter in Norwegian, or healthier goats. So the funding was governmental, but also from the industry, and also partly also from the farmers itself. And the goal was to eradicate not only CAE, but also caseous lymphadenitis and paratuberculosis. I just talk about the CAE. So at the start, it was voluntary. There were two methods: either stamping out of seropositive animals in herds less than 10% CAE positive. It's a test and cull method. Then the second one is snapping of newborn kids. And these kids were isolated in new building and given co-colostrum and milk replacement. And they were tested several times already when they were young, but also later. And the old flock was then slaughtered at the end of the lactation period, and the building was washed and disinfected, and the so-called non-infected goats were then moved into to the barn. And these herds that was enrolled in the program was tested regularly for a 5-year period. So the industry wanted all the farmers, or the dairy goat farmers, to be involved. But before 2012, there were several farmers that wasn't involved in this system. So they decided then to lower the price for goat milk from these herds. So within half a year, all dairy goat herds were in the project. And here you see also a table looking at the two methods, snatching or test and cull. And if you see the result here, 87% of the farmers choose to have snatching. So the serum test they used the ELISA from IDVET, and for inconclusive and positive results, they did it again in duplicate, and again an inclusive or positive test, but further tested in an ELISA from IDEX, or earlier from the agar-gel immunodeficient test. So in case of positive and inclusive results, new samples were tested, not only for that animals, but also from the herds. So the number of flocks that was involved here was 606 herds, mostly dairy goat herds, but also from other breeds, hobby meat breeds and fiber breeds. And here you see the number of samples during the program that was analyzed. And here is from the goats, and here is from the sheep. And because this is sheep from mixed farms, they have both sheep and goats. So in total, it was nearly 140,000 samples were analyzed. So in summary for this project, as I said, 606 goat herds have been declared free from CAE, and the cost from the government were more than 5 million euros during this 10-year period. But there are a rest of small herds that has not been involved in this project, and the average herd size around 5 animals in those herds. So there were some improvements that the farmers observed in this flock. Seems to be healthy animals, the increase of welfare, more sustainable animals, and also increased milk yield and improved milk quality. And here you see an example that's indicating that after snatching, especially from the third and fourth lactation, there are increased milk yield, indicating 40%. And when it comes to somatic cell counts, you have a similar or a reduction of 150,000 cells after snatching. So CAE has been, in 2012, it was a category B or list 1, mandatory. So then again, it was a surveillance program. So this is the restriction then, restriction of movements. So the movements of those is forbidden. So if you're going to move female goats, you have to apply for exceptions. It's very restricted. And all bucks, if you're going to move them, you need a health certificate from a veterinarian less than 7 days old. And what about genotypes? I just mentioned that. From the goats, it hasn't been mentioned earlier, but it's goats in Norway are in type C, while the sheep are also in type A. But to look at more detailed results from Norwegian isolates, you see sheep are in group A, but also in group C. Most of the sheep in Norway, at least in the recent years, are found in the group C. But all the goats are found in group C. I don't have any good explanation for that. So this is what's happening now in order to try to avoid infection with CAE. You have the surveillance program with individual blood sample. It's risk-based. And here you see the total number of herds in Norway. And also here you can see the number of positive herds. And in 2022, 1,800 goats were tested and none was positive. But that is the national surveillance program, but the industry has itself a program. They take both bulk milk samples 4 to 5 times each year. And if they are positive, they go further. So the number of positive animals in Norway, you can see here, it's from 8 to 0. And most of these 5 or 8, this is from meat, fiber, or hobby farms that has been starting to try to eradicate that. But there are some farms here that has been declared free, that is positive again. And there are some indication in Norway that infected goats may seroconvert after 7 years. What about reason for eradication failure? As I recently mentioned, the delay in seroconversion is one explanation. It could be management failure, and not all farms are involved. And could we avoid illegal transportation? We don't know. But it's still necessary to have this surveillance program. So you have these individual blood samples, and we have these bulk milk samples. So And I said, if this is positive, we go further with milk, individual milk sample, and then further again. Yes, I think that was the whole short story.
Thank you, Snorre. It was so short story, but well, nice story, you know, because you are, well, yeah, a long story also, yeah, but also a nice story because, well, you succeed with eradication of the disease. Well, you just mentioned that you have had a case of seroconversion after 7 years. Is it true?
Yes, yes.
Well, well, we had such a experiment. We kept, I don't remember how many, 20 or 30, something like that, 30 bugs together with infected mothers for 3 weeks, yeah, all the time. And then we were checking the serologically those animals for 3 years. And how many, Michał? 30% converted? 30% of them converted. So during 3 years. So I think it just supports your information. This is also one, well, bad thing about the capronarthritis encephalitis. The seroconversion is simply long, or maybe we should say very long. Yeah. Well, you didn't. Well, you find that the huge influence on the milk performance this infection. We didn't. Well, again, this is just the disease. Still, we have to do some research on it. Zosia, are there any questions? Not any. From the audience.
Thank you very much, Snorre. Impressive data. You showed a really impressive increase in milk production in the goats that were sanitized by CAE. How much do you think is the contribution of the small ruminant lentiviruses, and how much is the contribution of management improvement and get rid of the other pathogens that were also in the earth? Can you speculate on that?
I think it's part of it, but which part or how much, I don't know. But there are also some indication that, for instance, Mullerius capillaris, lungworm, in this flocks that has been got rid of CAE has less infection. So I think it's several things contributing to that result. So not only CAE, but that's a part of it.
Well, the long story. It's about the cattle, not about the small ruminants. Once we did really a very well detailed experience with calf diarrhea in one herd. So we vaccinated some animals. We did not vaccinate the cows in another farm, and then we try to evaluate the vaccine, and then we find out that the most important factor, it was just the people who work with those calves. So it doesn't matter this vaccine, but if those calves really were well treated, they got the colostrum properly. So vaccine, of course, helped, but this people factor was the most important. I don't know how it is with the goats in Norway, but well, yeah.
I think it's the same. The manufacturer and the management factors are very important. It's more interesting people that are involved in this, has been enrolled in the project. So I think that's a good part of that. Yeah.
That's also the answer for Giuseppe's question. Yes, as always, it's not so easy. You have to take a lot of factors together. So then try to get any conclusions. Any more questions? Not any. Thank you very much, Snorre. Thank you very much. And let's go to the next one presentation. Giuseppe, we are really happy that you came to visit us. And well, the floor is yours.
So thank you very much, Jarek and Michal, and all the teams for the organization of this very nice meeting. So let's go to the Swiss story, which is similar and maybe not as successful as the Norwegian one. So are we free? Are we not free? And I will do a little bit an historical summary of what we experienced in Switzerland. This is the situation now. We have about 82,000 goats in Switzerland. You see, as all the countries, you go to the if you go to the homepage of the Swiss Goat Breeding Association, they will tell you that Switzerland is the country of origin of modern goat breeding. Probably at least 10 countries are claiming the same. And the problem is what is Switzerland exactly? This is not easy to explain. As you probably know, the geography, Switzerland has these strange maps, very stable, neutral country, but we have some movement. Just few months ago, one city of the canton of Bern decided to go to the canton of Jura, and we managed to do that without a civil war. So it was quite an achievement. But 200 years ago, Switzerland was looking differently. The color is the real Switzerland. I'm coming from this corner, and you see it was not Switzerland at that time. The valley, very close to my valley, also Italian speaking, was taken over by the Swiss-German canton. So we were, it was, this one was really Switzerland, this not. This was a colony, and they were making every year, the Swiss-German cantons, they were making an auction, and the one that paid the most was getting the old canton of Ticino in the south, and for 4 years he was able to collect taxes there. But what has that to do with goats? We are in the south one of the cantons with the highest number of goats, and this is, we are very proud of this breed, the Nera Verzasca, and most of these goats, they were not in Switzerland at that time. The same with the canton of Graubünden, Grigion. Those, they had this Capra Grigia and other breeds. They were not part of Switzerland. The real Swiss goats were the Sanen, that you know, the canton of Bern, and Toggenburg, and somehow in the valleys they have also goats, but they were not part of Switzerland at that time. So change historically, you have a lot of changes. And to explain what happened with caprine arthritis encephalitis in Switzerland, I'm using Dante Alighieri, the big poet that told the story from the paradise to hell, and I will start to hell and try to go toward the paradise. And let's go back in the 80s of the last century. What we have, we had really the farmer that were weeping, especially the Goat Breeders Association. They were desperate. The goats were in a very bad shape. They have less milk, they had no cheese, and they were surviving on cheese, and they were losing a lot of animals. And the pressure came from the Breeder Association that went to the university and said, what the hell is going on? Can you do something against it? And some people were involved, very important people. Peter Ammann was an agronomist with very good contact with the breeders, and he went right away to the virologist, Peter Ants, Sandor Lazari, I think from Hungary he was, a very nice person, and he was an genetic immunogeneticist, so genetic and immunology. Claude Gaillard, also a geneticist. So all people that knew about genetic and that were right away, they understood that this is a problem that has to do with genetic. Gabio Wechsel-Ruf, she did a fantastic work over the years. Reto Zanoni was also involved, and also practitioners like Joseph Regli were at the beginning. So at the beginning, whereas the constellation, the Goat Breeder Association, plus the people in the university and in the association that wanted to do something. And you have to start somewhere, and they started in this little village in the corner of the canton of Graubünden, and they started a pilot project, and it was in '84, and the breeders there, they wanted desperately to start something. This was a big success, and as it works, the news spread. Other breeders were convinced that we have to do something. They had a fantastic success in Andere. And so slowly the things start to move. And it was really a bottom up and not a top down approach that worked. So in 91, you see almost 10 years later, finally they realized that this loose association of goat arthritis was not sufficient. So it was from the state and was with the financial aid of the state. Finally, they started this advisor and health service for small ruminant. Raymond Misery is the head there, and he's still working at the beginning only on small ruminant lentiviruses. Yet now they do a lot of different diseases. Then we had finally at the level of legislation in '95, it was decreed by the federal authorities that this is a disease to be eradicated, which means you have money, you can compensate the farmer, you can really start a program. And you see, it took 3 years from the decision you have to eradicate to make the whole story compulsory. And if you look at the map of Switzerland, more and more farmers, they were participating. And the whole process was followed by the Institute of Veterinary Virology till 2013 with Ernst Peterans. He invested a lot of energy in that. Reto Zanoni and also Hans Rudolf Vogt was the head of the diagnostic department. He did a fantastic job with this disease. And in 2014, we fused into the Institute of Virology and Immunology. And for CAV, it was again Reto Zanoni and Carlos Abril that were carrying on this work. And since 2022, it is Carlos, the head of the diagnostic for small ruminant antiviruses. These technicians were seminal for the success, especially Van. She worked 40 years at the Institute and many years just to fight this disease. And the last two people that I would like to mention is Elena De Martin. She helped a lot in this last year. And Maxim, that came from Ukraine or from Poland, from Yareklad, and he came to us, and he was very involved in this end of the program. And 2019, finally, we had the triumph. We got rid of the virus. Did we got rid of all the viruses? Not really. You see, this is the official document in German or French, but obviously the meaning is we got rid of these CAE viruses, but the situation is a little bit more complex. Let's go back to a very important work of Beat Thoman, which is an epidemiologist, and he helped us to evaluate with his group, to evaluate the results of a census analysis that we did in 2011 and 2012. There we look at every single goat in Switzerland was sampled, 85,000 and something. And what we found was very nice results. Genotype B, so the real CAV virus, we found 47 positive goats distributed in 41 flocks. So it was almost one goat per flock that was positive, very low seroprevalence. And if you look at the map, this is not an infectious disease. It's just in some flocks somewhere in Switzerland, you have one positive goats. We thought this is no longer an infectious disease. We got rid of this. And Concerning genotype A, the mydivisna type of viruses, situation a little bit more complex. We can say about 10 times more positive animals. Very low seroprevalence, but is there. The virus is there. And if you look at the map, you have the feeling here you can have kind of clusters, and we had more than one animal positive per flock. So here we had the feeling we still have something going on. And our conclusion was, I always use the symbol of the hydra for these viruses. And if you take one of the most recent phylogenetic trees of this virus, is really a hydra with an incredible number of heads. And what we think for Switzerland, we chopped away the B1 head of the hydra. That's the one that was inducing this type of disease. We will discuss that because Jarek, I know he doesn't agree with it, but we will see. It's in our experience, getting rid of this virus got rid of the arthritis, and this one must not grow back. We had a problem with importation of goats from Belgium that were all positive, and we were lucky to be able to act very quickly and to stop the spread of this virus. Those heads of the hydra are a little bit more complex to fight, especially the A4. Those are dominant. I don't know why in the Swiss population, especially in sheep also, we have these viruses. They make the serological detection is sometimes difficult. We had tests that fail with A4. We have cross-reactivity with the B test, and we invested quite a bit of energy to figure out, are they virulent? Do they induce a disease or not? And I will not talk about that, but I can tell you, not the classical disease, but they tend to go into the mammary gland. And how many head of the hydra we have? We don't know. And it's getting more difficult because the money to invest into these diseases is drying out. Now, what we did in the last year, basically, we decided after 10 years to go back to the flocks that had some of these A viruses and just look, did it spread, did it not spread, why it's spreading or why it's not spreading. And this is a work of Fadri Vincenz. He called several of these farmers, and because it's no longer compulsory to do something, a lot of the farmers, they say, thanks, no, if you can stay away from us, it's better. They didn't want us to go in and test the animals, which is understandable. We found 7 flocks that agreed to do that with 145 samples. Fadri, which is one of the most experienced veterinarians that I know working with goats, he did a fantastic clinical examination. He took EDTA samples, serum samples for PCR and for serology of 145 goats, and the results are quite encouraging. We were terrorized by the idea of finding a B positive goats. We couldn't find them. They were bad. In all the more than 100 animals we found, they were all perfectly negative. In one herd, in the French-speaking part, we found that 8 out of 13 animals, they were positive. They were positive by PCR, they were positive by serology, but they were of the A4 type, so the type that we know it was around. Clinically, nothing. They were perfectly healthy. When Fadri told me that, I told him, are they living with sheep? Because we have experience with it, and we know that the virus is jumping very easily from sheep to goats. And I told him, that's clear, 200 sheep, and that's where the virus is coming from. But it was wrong, because the farmer was a sheep farmer, very careful in the examination of his sheep. They were perfectly negative. So what happened in this flock, the farmer kept the goats as they were in 2011, and the virus spread over 10 years without inducing clinical disease, but it's there. And that's about the situation. Very easy to summarize. We are bee-free, but don't ask too many questions about the A because they are circulating and they are in some farms. And again, Michael and Jarek, thank you very much. And for you, thank you very much for your attention.
Giuseppe, thank you very much. And well, you only add some evidences that this is really strange disease and not so easy. Yeah, not so easy to eradicate. And well, it needs maybe many more time and many more efforts just to do something. But maybe somehow, well, it's nice that we have such a disease because still we have something to do, you know. Okay, thank you very much. Just any questions, Zosia? Not any question. What about the audience? Oh, there is one question. Okay, Snorre.
The revenge of Snorre.
Thank you very much. What about type C? Do you could explain why we only have that in goats in Norway?
I think this is really an historical fact. Probably is something that is limited to the Scandinavian country. I don't think that they found C in other countries. I know from Sergio Rosati in Italy, they have the type E that they found in the Roccaverano breeds in the Piedmont. And it's really only there, it's local. This is a very fascinating virus, the type E, because it's apathogenic. It has mutation in the genome that make it less virulent. And you find it in the Piedmont, you find it in Sardinia, because it was part of the same kingdom, but you don't find it anywhere. And probably you are the only country in the world that has the type C. Maybe with Sweden. I don't know Sweden. Yeah.
Okay, thank you very much. Thanks once more, Giuseppe. And now the last one talk from Michał. So Michał is the head of the project I was just telling you on the very, very beginning. Michał, now it's your time. It's yours, yes.
Thank you very much. Okay, so I will try to say a few words about the project, which is the reason for which we are here. I mean, the disease is the reason, but the project is our idea to somehow deal with this disease. And let's start with a small idea which I have, because we've been investigating many caprine diseases here in Poland for last 15-20 years, Professor Kaba for 30 years. And I think there is one conclusion which we can draw from all these epidemiological studies and all our knowledge of this situation. And this conclusion is that Poland is quite a specific country. I don't mean that there are no other such countries. There are many such countries, but this is a specific type of a country in which we have quite many goats. Let's say it's not few. We have more than 100,000 goats. Yeah, so it's quite a lot. In really many herds. Most of them are very small, but let's say that there are herds which are developing and herds which are trying to get better and better and be more profitable and rear kids and keep goats in better conditions. These herds have problems because, let's be frank, that farm animal veterinarians let's say pay no attention to goats, and they don't know much about goats, because goat is not a target in Poland and in such countries. Even more interested in goats is the state, I mean government, which is not about to give any money to control diseases or do something like that. As a consequence, in our opinion, the only way to keep any disease under control in such a country, in such a population, is to give farmers and veterinarians a very simple tool which will be extremely easily available and will allow to make a diagnosis. because any other method which requires, for example, sending samples to laboratory and waiting for results, any method which is expensive will simply not work, not because of the fact that it's not sensitive or specific or anything like that, because probably they are very good, for sure they are very good, but they will not use it. And this is from where our idea came, yeah, to develop the test, which will be just an on-site point of care test and will be available for not only for veterinarians, but also for farmers, because it will not require collection of the material, which, by the books, can be only collected by the professional veterinarian. And this is where this idea for on-site serological diagnostic test for caprine tratis encephalitis came. So we applied for the grant from ICRAT. Here is the full name. I'm not going to read it. And this is quite a specific grant because it's realized by the consortium of 5 countries, but each country is financed by its own agency, national agency, and gets money directly from this agency. These agencies are also listed here. And these countries which are involved is, as we've already said, Norway, Switzerland, Hungary, and Lithuania, and Poland is the leader of this project. And what we're trying to do is to develop the test. And let's say that caprine tratis encephalitis is an example of the disease which is transmitted between herds with an animal. I mean, it's very difficult to spread this disease in any other way. You just have to move an animal from one herd to another. And there are 2 basic situations in which something like that happens. The first one is when the farmer wants to develop the herd, wants to build it up and have it bigger than before, has to buy animals from somewhere. And in Poland, for example, buying animals from somewhere usually means that you have to buy them from some very small herds. Either you buy these small herds or you buy single animals from many herds. And you know nothing about, and the farmer, the one from whom you buy it, knows nothing about their real health status, yeah? And you just gather them from everywhere, so you're gonna get everything, yeah? So this is the first high-risk situation. And the second one is that the problem which we have with artificial insemination, let's say we don't have problem because we don't have artificial insemination in goats, yeah? So if you want them to breed, you need a male, but there are much fewer males than females. And the another thing is genetics. You need diversity. So males have to travel from one herd to another just to ensure this biodiversity and genetic diversity. And it's a nice mode of transmission of the disease. And how we can try to overcome these problems. There are 2 solutions. Yeah, one solution is the solution for rich ones. So this solution is, let's close the herds. Let's lock them up without any access of any new animals. Let's just rely on our own replacement. But to do this, we need artificial insemination, yeah, to create such a herd, because we need genetic diversity. This is something what is very difficult, what is laborious, what is most important, very expensive. Yeah? So the other way is to test goats before we let them enter the herd. It's also for rich ones, because not only because of the fact that tests are expensive, because some of them are more, some of them are less expensive, but to do the test we need to collect blood from each single goat. It has to be collected by a veterinarian, sent to the laboratory, and you've got to wait for results several days. And during these days, this goat has to be somewhere, yeah, and you have to do something with this goat. You cannot let it have contact with other goats. So this is also the solution for richer countries. That's why we think a good idea is to develop the test which will be easily available for the farmer. So it will be just present on site. And if the farmer wants to buy a goat, just takes this test, collects material, which will be milk. That would be the best idea because you can easily collect it. However, it has limitations, especially in males. Yeah, and or the blood drop, and you just test it and have results within 10 minutes, just like it is in many other animal species. So it seems very simple. Yeah, it sounds very simple. It's a simple idea. There is one important thing which we have to remember about such a test is going to be a screening test. Yeah, so we can be sure that it will not be equally sensitive and equally specific. So we have to choose something, and we decided to choose sensitivity because we want to be as sure as possible that when the goat tests negative, it's really healthy. It's what we really want to. We want to trust negative result. If there's negative result, you let this goat in. If it's positive, you need to test it further. Maybe in fact it's just false positive result. Maybe it's true positive result, but let's better stay away from this goat. And that is our idea. So this project is made of several stages, several phases, let's say. And the first one was development of antigens for this test, which is fortunately already done. We have a set of antigens. There are three main antigens which we have. One is recombinant antigen of the capsid protein. The second one is the surface glycoprotein, of course only the part of the peptide from this glycoprotein, and transmembrane glycoprotein, of course also a peptide. These are antigens which come from goats from Poland, from those viral strains which circulate most commonly in Poland. They are A strains and B strains. And we know that they work in ELISA, so they work in laboratories, and now they are about to work in a rapid test. So now the test is being manufactured by the company. There are, of course, many various problems with changing this test from ELISA to immunochromatographic lateral flow test, because it's not the same, and these antigens have to be modified to work in this type of test. However, this is something what is going on now, and this is done by a subcontractor in this project. And when we already have this test, we will test it in laboratories to prove that its sensitivity is really as high as we would like it to be. Oh, for sure, much more than 90%. Yeah, the higher the better, but it has to be higher than 90% to be considered a really trustworthy screening test. And we will try to test it both on symptomatic goats and asymptomatic goats, somehow to reflect our idea that they will be used in animals with clinical signs to check whether it's caprine-tritis encephalitis or not, because the same clinical signs may be observed in other diseases as well. And also in asymptomatic animals, which usually happens. We have an asymptomatic animal and just want to know whether it's infected or not. And we will focus mostly on sensitivity, but of course, we hope specificity will also be acceptable. Yeah. So that would be good to have it also higher than 90%, but we will see. When this test proves effective, accurate, let's say, in laboratory, it will be tested in field conditions by our partners from Hungary and Lithuania, in herds in which the presence of the infection is at roughly 50% level. These animals will be tested by veterinarians who know nothing about the real status of these particular animals, just to let this part be blinded. Yeah? and samples from these animals will be sent to laboratory, and we'll have cross-check of the status of these animals using our rapid test and laboratory tests. And this will, let's hope, prove that this test really works and works very, very well. And we hope that the emergence of such a diagnostic method on the market will make farmers and veterinarians more aware of the existence of this disease, because this is usually how it is in veterinary practice. When we have a test which is easily available, we become more aware of the existence of such a disease, and we hope this test will somehow bring this disease to as something more easily recognizable for farmers and for doctors. And of course, we hope that it will also increase the welfare of animals. And that's it. Thank you very much.
Questions from the audience? Meron, just a moment.
Now is the question from field practitioner. So
I have questions. Why you choose milk? I understand obviously that blood is quite difficult from the owner to obtain, but for example, why not saliva, which is quite easy to obtain by owner? And all animals have youngs, males, females, everyone have saliva, okay? And it's quite difficult, quite easy to take saliva. So why you didn't aim this material in your method in order to?
I think the simplest answer is that if milk is about to kill this project, saliva for sure will kill this project, because finding antibodies in saliva and preparing the test which will work on saliva is on its own so horribly difficult. And this is about to be the first rapid test for caprine-enteritis encephalitis. We wanted to work on blood first, then hopefully on milk. If it works on milk, we will be thinking about some other materials, biological materials. Your idea is a very good idea, because everyone has saliva and it's easy to collect it. But it's very, very difficult to develop the test for this material. And that's why we haven't chosen it.
And Meron, we even have had a better idea, because we did some studies on saliva. The level of antibodies in saliva was so low that, well, as Michał said, let's wait. But we had also, Michał, you should tell what we also did, because just to collect the saliva technically from the goat. But we also elaborated the method just to take it. So nowadays we know how to do it, and really it works. But still we had a crazy idea. Why saliva? Maybe there is some other material which we can collect.
And everyone has it.
Yeah, easy to collect it. Feces, why not in feces? We even checked it. Doesn't work. Sorry, I have to pass you the microphone, because otherwise nobody will hear you. So.
I think that the problem can be related with a lot of inhibitors in feces and in saliva, and also milk is more predictable sample. It's usually always the same. And saliva is always different. Feces can be also always different regarding the composition, same saliva.
I fully agree.
Yeah, but Michał even tried, to with the feces. Yeah, he tried to prepare them several ways. just and checked with the ELISA. Didn't work, unfortunately. So, but this is maybe the future. This is maybe the subject for the next project. You know, just to try to change, because it will be really easy to take the saliva, as you said. It's a wonderful idea, or to take the to sample we just thought if it works with the feces, so we could do both the parasitological research and at the same time the CAE. So that was our idea. But you know, the ideas are nice, but when you come to the reality, well, it needs time, it needs work. Maybe in the future, maybe next project. Kinga, it's a subject for you, maybe, you know. Okay, so any other questions? Okay.
Will the rapid test also be tested in herds in Poland?
Yes, it will. I mean, first, so it will be tested on samples from Polish goats and Norway, goats from Norway, because they are negative. Yeah, so it's a very good source of negative samples. And then the test will be evaluated in herds in Hungary and Lithuania. And afterwards, of course, it will be tested also in herds in Poland. Yeah. So and we hope it will be tested in many different countries when it's already on the market.
The idea of testing it first in Hungary and Lithuania, it was, as Michał already mentioned, a blind study. So the idea was that we will send them the ready test, and then they will go to the field, test the animals, and then they will have the results of the test. Then they will send to us the serum samples, and we will do the serology. Everything will be blinded. This is the way how the accuracy and of the test should be studied. So that was the idea of this first trial done in Lithuania and in Hungary. That's why it has been planned like that. But if the test works, for sure we will use it in Poland, for sure. And if it works, well, there is ID company in here, so maybe they will produce it, and maybe it will be available all over Europe. That's a dream, you know, but always you have to have a dream. Let's hope it will be like that. Let's hope it will end like that. Let's hope. Well, anyway, everybody in this team works on this, and Giuseppe has produced already the antigen. The antigen is now in the productive plant here. They are just working on the preparation of the ready of the test. Snorre has supported us with the negative samples. We have the samples from Lithuania and from Hungary. Let's hope the test works, and if it works, so everything is already ready just to assess the test. And let's hope it will be wonderful, and let's cross the fingers, Michał.
There's one important aspect of this question, which has just been asked, because the test is developed on the antigens from Polish viral strains. Yeah, in Poland we have goats from whole Europe, let's say, but do we really have virus from whole Europe? We don't know. Yeah, so this is an interesting part to compare how well it works on Polish goats and on goats from other countries. And let's be frank, usually it is like that it works better on the population on which it has been developed compared to working on, you know, other populations. And we are aware of this fact. But let's be also frank that most of companies don't check tests like that. You know, this field checking on other populations is not typical. We just go one step further. Of course, it's a risky step. Maybe it will turn out that, okay, in Poland it works, you know, sensitivity is very high, you know. In Lithuania, it's 70%. Well, we have to face it. Yeah, that's science. That's why it's a scientific project, not you know, not industrial project. Yeah.
Yeah, but if the test is really produced, if it really works, so then just change the antigen would not be such a huge thing. Yeah, just to produce the antigen from another strain of the virus. Yeah, the most important thing is just to establish this test, as that's what we are trying to do now. Later on, it's a lot of work and a lot of opportunities for further improvement of this test. Let's hope that it works. Let's hope that our work is not for nothing and will give us some fruits on the end. I trust that we will succeed. Okay, any other questions? Any comments?
So the antigen for this rapid test are from genotype A and B from Poland. It cannot be a problem if we test goats from Norway, which have type C. I don't know, I'm just asking, it can be a problem when we will test samples from Norway using this test.
But you mean that the problem would be that they are false positive in our test?
No, because in Norway we know that we have negative, but imagine that in Norway we have also positive goats.
But can be C positive,
C positive,
and as a consequence, our test overlooks them, misses them because it's not based
yes,
on.
this was the question. If you think that this can be a problem.
I think C genotype is quite similar to A, but it's just like, answering all the questions which we theoretically answer. Maybe you're right. We'll check it.
Okay, yes. Any other questions? Any other questions? No questions from internet, not any? Okay, Michał, thank you very much.
Thank you.
Thank you very much. And thanks for the presentation to Michał, and thanks to Michał for being with us so many years and collaborating with me. I think, well, I don't count the years already. So, okay, I think we are close to the end of the conference. So once more, I would like to thank to all the lecturers. It was really a big pleasure for me to host you here in Warsaw, and I think that the conference gave some. Well, everybody of us have had some other point of view. Well, because the disease, as we said, is complex and not so easy. And then the other thing is that there are lectures coming from really different countries, Switzerland and Norway, and really rich countries. So you have a totally different possibilities to eradicate the disease. And well, let's say Poland and other countries, the financial sources, they are much more lower. And that's why we thought about this. That's why we thought about establishing this new test, which should be really cheap. And well, when we have been on the conference in Sevilla, in Spain, somebody asked us what for to develop this test, because it is already the PCR test, which works very well. So what for to elaborate the new test? Well, the question is simple, yeah. Okay, if you go to the farmer who has 5 goats, like Snorre said in Norway, and it's the same in Poland, and this farmer would like to buy a new goat, to introduce one goat to his or her farm, and this farmer lives somewhere in Poland or in Hungary or in Lithuania, and you will advise as a veterinarian, advise him, okay, now let's take the blood, let's send it for the PCR to the laboratory and do all these things, and then pay for veterinarian for doing the tests. So in our situation, it's not a solution. The cost of all those procedures will be so high that the farmers will say, let's forget it. And we would like to do something against it, because we have seen how the disease has been spreading in Poland. It was just this reason. Nobody was testing because it was too expensive. And from from 25% the prevalence, which was in 1996 when I did this study the first time. Now we have something like 70-80% of prevalence. So we would like to find a tool which will be cheap, which will be reliable, and we hope that this tool in the future will just help to stop spreading the disease. Let's hope it will be like that. Okay, thank you once more for everybody. Thank you for our university TV because they just allowed us the whole transmission. Thank you to Zosia and to all, to Marcin and to Agata and to Kinga and to everybody from our team. Thank you because well, without their help and without their support, everything won't be so easy like it was. So thank you very much, and thank you all, everybody somewhere there sitting at home and watching the computer. I hope you enjoyed the time, and I hope you got some new informations and some new ideas. And thank you very much once more. And well, let's maybe see once more few years later for the next conference when we will already see the results of the test, Michał's test done in a field all over Europe, let's say like that. Thank you very much. Thank you.